like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Lin School of Medicine Healthy Longevity webinar. It's great to have you on board tonight. Today's focus is on cellular senescence. It's a very important mechanism for disease incidence and progression in humans. And Professor Cleo Bishop is sharing her latest results. You might have lots of questions, so please use the Q&A function to send out your comments and the questions, and Sharaka Pimping will moderate the Q&A session today. We start with a short presentation by Prof Theo. He is our intern from the Temasek Polytechnic, and he will talk about his own project in identifying novel factors and skeletal muscle cells after exercise in women.
Thank you for the kind introduction. As mentioned, I will be talking about my major project during my internship for Center of Healthy Longevity at NUS. The title of my project is Identifying Novel Factors in Skeletal Muscle Cells in Response to Circulatory Factors from Young and Old Women After Exercise. So what are skeletal muscle cells and circulatory factors? Well, skeletal muscle cells make up one of the three main types of muscles in our body, and they are commonly found attached to our bones. We also have the ability to control these muscles voluntarily. As for circulatory factors, they are also known as myokines, and these are molecules that are responsible for the communication between cells. So how are skeletal muscle cells affected by exercise? A study conducted in 2005 observed that participants who performed Katsu, a form of resistance exercise, had an improved skeletal muscle cell diameter. Additionally, they also observed an increase in a myokine called insulin growth factor 1 among participants who took Katsu compared to the control group who didn't. Singapore has a growing problem of an aging population. Pair that with a low birth rate, and there is a significant urgency to research and develop effective methods of intervention to help prevent Singaporeans from experiencing premature frailty. However, there are major gaps in our understanding and knowledge of which types of exercise help improve the production of these beneficial myokines and prevent aging. To make things worse, many documented literature from different groups also have conflicting results. Therefore, we hypothesize that one type of exercise will cause certain kinds of myokines to be produced compared to another kind. This is a brief overview of our experimental design. We will conduct conditioned media experiments using mouse myoblasts at various Besage numbers and grow them in human serum conditioned media obtained from healthy individuals. Then we will assess the cell's sensitivity and interactions between themselves and the growing media through visual inspection, cell counting, and ELISA. Using the results from these experiments, we hope that the novel myokines and biomarkers that can be identified in this project will serve as a stepping stone to accelerate gyroscience research and improved human longevity. Thank you for your kind attention.
Thanks, Raif, and thank you for your contribution to the Center for Health Longevity. We are really looking forward to your results. But now we will go to our prime speaker, and that's Cleo Bishop, and she is a professor of senescence at the Queen Mary University of London in Europe. And I know her from her work within ICSA, and ICSA is the International Cell Senescence Association. Her group is studying the role of senescence in aging and in cancer, and she loves, I know her a little bit, she really loves to have a deep dive into mechanisms of cellular senescence to find anti-aging and anti-cancer therapies. And we are fortunate that she is not only a very great speaker, because I have heard her a couple of times, but she will also share some unpublished data which explore how DNA methylation clocks can be harnessed for drug discovery, which is so important. So Cleo, my first question to you, is it tick-tock or tock-tick? The floor is yours.
Thanks, Andrea, and thank you ever so much for the invitation to share with you our unpublished data today. Hopefully you can see my slides. So yes, thanks again for the invitation. It's a real pleasure to be speaking to you from London today. I'm actually sat in this little white blob in our institute, which is our mitochondria as designed by our architects. So it's a pleasure to be speaking to you today. And as Andrea mentioned, I'd like to share with you some unpublished data that we're really excited about. And I'd really welcome any thoughts, any feedback, any critique of the data, particularly, as I say, as it's new. Also, if anything I show you today piques your interest, we're very open to collaborations and to hearing from people who might like to work alongside us with some of the things that we're investigating at the moment. So as Andrea says, is it tick-tock or tock-tick? And maybe the title of my talk might give you some clues as to where we are hopefully heading today. But before we get there, I thought it might be helpful, and I'd be grateful if you could indulge me with a little bit of context. I'm sure I'm speaking to people who know a lot already about the hallmarks of aging, but I just wanted to put a little bit of what we're going to talk about today into some context. So you'll all be familiar with this, the wheel of the hallmarks of aging. And today, what I'd like to really focus in on is just the three of these hallmarks. So epigenetic alterations, deregulated nutrient sensing, and as Andrea mentioned, my lab is particularly interested in the process of cellular senescence. And I'd like to take you back perhaps a number of years now to a curiosity that we had. And so what was this curiosity? Well, you'll all be familiar, I'm sure, with this schematic of the Hayflick limit, the fundamental process which underpins the cellular senescence program. And in my lab, we are particularly interested in cells that have reached this deeply senescent state, which is a state where the senescence program has been enforced, maintained for a period of time, and the cells really start to elaborate a full senescence phenotype, which includes the senescence-associated secretory phenotype, or the SASP, this cocktail of a myriad of different cytokines, growth factors, and so on, which are thought to play a really important role within the chronic state of senescence within our tissues and the local sterilized inflammation that these cells might contribute to. And so my lab, the curiosity that we had a little while ago, was really to ask whether we could take cells that were in this deeply senescent state, which had been in senescence for a period of time, and reverse this phenotype. Could we take them backwards in time? And we were wanting to do this very, very carefully, and we were very conscious in all the experiments that we did to make sure that we weren't driving these cells out of senescence into what we refer to as an escape mechanism, essentially driving them towards a pro-tumorigenic phenotype. And our kind of motivation for wanting to ask whether we could reverse senescence was really to see whether we could use this as a tool to understand in more detail the drivers of this fundamental cellular program. And so, as is always the way, when you think you've had a good idea in science, you look in the literature and you find out that actually others have had a similar curiosity in the past. And these are just some of the papers that were published a number of years ago, really asking a very similar question, whether senescence was truly reversible. And so these publications, which include data from Judith Kambisi and Masashi Narita, really encouraged us to pursue this question in more detail with more sophisticated techniques that were available than when these researchers were looking at this question about 20 years ago. And so this really did become what we refer to in the lab as a Friday afternoon experiment. I don't know if the PhD students in the audience still do those. I think they're fantastic. And essentially what we did was spend many Fridays trying to see if we could introduce sRNAs into these deeply senescent cells. And actually that was the trickiest, the most challenging hurdle that we needed to overcome to be able to ask this question. And so the data I'm going to show you at the moment is in adult human mammary epithelial cells, which undergo a very clean P16 mediated senescence program. And I highlight the fact that these cells are adult because we believe that neonatal cells, which a lot of people use in their research, have fundamental differences that I'm happy to chat about later on. So this is just a very simple set of images showing our young, early proliferative HMECs and our deeply senescent HMECs, which have been stained with one of the key markers of senescence, senescence-associated beta-gal, which we see both present in cells in vitro, but also cells in vivo, one of the two kind of most robustly used biomarkers of aging in vivo. And so essentially, many Fridays later, what we were able to do was to take these deeply senescent cells, and by introducing a range of different sRNAs, we were able to generate this reversal phenotype. And so hopefully you can just see from these microscopy images that the cells within this culture are fundamentally different. They've lost the kind of classic fried egg morphology that we associate with senescent cells. You can even see a mitotic pair within this image. The morphology of the cells is fundamentally changed. And so we went on to characterize these changes in a lot more detail. And so in addition to a downregulation of SA-beta-gal, we also see a loss of P16, the key effector and maintenance factor of the senescence program. We see a loss of these SAS factors that I mentioned earlier. We see a complete suppression of reactive oxygen species amongst many other markers of senescence that we examined. Really importantly, and as you can see from this image, the cells have re-entered the cell cycle as assessed by BRDU, and they have proliferated and have this range of morphology changes that I mentioned earlier. So these was really nice. We were able to show by a range of different markers of senescence that we had fundamentally changed the cells and to all extents and purposes reverse the phenotype. But what we wanted to do was look at something a little bit more global. And to do that, we teamed up with Barbara Rakyan, who's a professor in my institute here, and a really talented bioinformatician, Rob Lowe, who's now at Altos Labs in Cambridge. And what we wanted to do at the time was to look at DNA methylation. And when we did these experiments, we were able to look at the 450K array, which enabled us to look at 450,000 CpG sites within the genome to assess what was happening to the methylome, first of all, when we take the cells from this young proliferative state into this deeply senescent state. And so this is the results of that first kind of question, what happens to the methylome? And so if we look at sites that are differentially methylated, what we call these SEN-DMPs, between the young proliferative cells and our deeply senescent cells, we identified 725 sites within the genome that are significantly altered in these cells as they go into this deeply senescent state. And for those of you who are interested in the kind of the dynamics of the methylome, they're pretty much split between sites that are hyper and hypomethylated. And so the really key question for us, having kind of established that the methylome does change in this senescent state, was to ask what happened when we took these cells through our reversal protocol. And although this next bit of data that I'm going to show you is quite old, it still remains very close to my heart as one of my kind of my most favorite bits of data. We really didn't know what we were going to see when we did this experiment, but we were really quite astonished to see that this is what happened to the methylome. So when we took our cells through the reversal protocol, what you can hopefully appreciate by the shifts in these two tones of purple dots is that they've essentially compressed back and essentially reverted very much to a methylome signature that is reminiscent of cells many, many weeks prior to this experiment. So within a very short period of time, this reversal protocol is enabled to reset to many extents the methylation signature of senescent cells to reflect those of early proliferative cells. And so what this enabled us to do was then go and ask how these signatures compared to aging-associated methylation dynamics that you'll all be familiar with from a range of groups, including Wagner and also Steve Horvath's. And so what we found was that these Zen DMPs were positively correlated with aging-associated methylation dynamics, but actually they weren't as good as aging-associated DNA methylation changes. And that's not entirely surprising when you think about what we are doing versus the aging clocks. We're looking at cells, very clean, single population. We're looking at them in vitro, and we're looking at them in a very specific and defined context. Whereas in aging, what you're looking at is changes that are happening within a whole organism across time. And we think that this is actually quite important for what we went on to do next. So we went on to publish this. So if you want to read a little bit more about what we did, you can see this in this paper from Lowe et al. And we then took a kind of a more challenging approach and looked in fibroblasts, which undergo a much more complicated and strongly enforced senescence program, which is mediated by both P16 and P21. And through a lot of work, Ellie Tyler was able to use our various techniques to reverse senescence in adult fibroblasts. And she used this to uncover a novel transcriptional regulator of senescence, the transcription factor EGR2, which sits upstream of both P16 and P21 to influence the expression of these two really important cell cycle dependent kinase inhibitors. And this work has also been published. So I'd like to move on to our unpublished data now and take you through what we've done through actually a really excellent collaboration across 3 different disciplines, I would say, to have this story for you today. And so this work has been done in collaboration with Cecilia and Ivana, who are both at UCL. Rob Lowe, whilst he was here with us, a really talented postdoc, Ellie Tyler, excellent PhD student, Ted Milligan in my group, and then with 2 of Stefan Beck's lab members, Amy and Simone. So this is very much a collaborative project working across a range of different disciplines. And so what we wanted to really ask was, we've kind of looked at this reversal process in cells in vitro, and we now know that there's a range of different genetic interventions which can actually extend lifespan in vivo. And so we know a lot about the aging clocks and how those are influenced during aging. And we also know that if you influence glucose sensing and a range of other things in yeast and mammals, you can actually influence lifespan. And obviously there's a whole plethora of ever expanding publications looking at the clearance of senescent cells, either through genetic interventions and also through senolytics, which are showing health span and lifespan improvements. So all of the data looking at genetic interventions really suggests that aging is a malleable process, something that we can intervene with to try and help us not necessarily live longer, but be healthier for longer. And so this kind of got us thinking about pharmacological interventions. And so when you look in the literature, there are some pharmacological interventions But really there's not as many as there probably should be, given the breadth of genetic evidence that we have that aging can be influenced. And so one of the kind of classic pharmacological agents that I'm sure you're all familiar with, so I won't dwell on this too long, is rapamycin. So this has been shown to extend lifespan in a whole host of organisms. It's also been importantly shown to extend health span. And it's been shown to delay the onset of senescence, to slow the aging clocks that we mentioned before. And so really what we wanted to do was to ask whether we could pharmacologically intervene with a clock of relevance. And so when we were thinking about this, you'll remember I mentioned that our senescence methylome showed some positive correlations with the aging clocks, but it really wasn't as compelling as you might have hoped for. And actually, when you think about it, it's not entirely surprising. We have a whole host of different independent factors which can influence aging and therefore potentially aging clocks. And so what this led us to think was that the aging clocks are actually taking each of these different hallmarks, blending them together to generate this delicious looking, but perhaps slightly muddled chocolate smoothie. And actually the individual hallmark signatures get lost in this particular approach. And so what this led us to ask was whether we could, rather than getting a kind of a chocolate brown clock, whether we could look specifically at a senescence clock. And by taking one individual hallmark of aging, would this give us a new route into exploring pharmacological interventions that could look to delay or influence aging? And so what we then wanted to do, this was a little bit later in time, at a point when the EPIC array was available, we wanted to ask what actually happens to the DNA methylation dynamics as we move from cells that are in a stable exponential growing phase. So before they've really got old and senescent, when we're in this period where they're still able to divide and haven't kind of obtained some of the features of senescence. And so we essentially profiled our cells at very regular intervals across a series of weeks and took these cells forward onto the EPIC array, which enables us to look at around 850,000 different CPG sites within the genome now. Really importantly for this work, we did this in the absence of antibiotics. There is evidence in the literature that antibiotics can actually directly influence the methylome. And so this was something we wanted to ensure that we weren't doing inadvertently. And so what we did, having done this profiling, was we then asked how well a range of different published clocks performed at predicting the change in time of our cells. And so we looked at a range of different clocks, but I'm just going to show you data for 3 of them today. So this is the multi-tissue clock, the skin and blood clock, and also the pheno-age clock. And so essentially what we found is that they weren't very good at predicting time. There's a sort of, some of them there's a kind of a general trend in the right direction, but actually they didn't perform particularly well. And that's not to critique the clocks. I think each of these clocks have got a really valuable position in a particular context, but in the context of tracking cells through time as they approach senescence, these clocks are not built to ask that question. So what this led us to do was to build a new clock. And we've done this work in a range of different cell types, all within adult cells, so both in fibroblasts and in fibroblasts from 2 different tissues. And we have generated independent data sets to generate our clock and to validate our clock. And to cut a lot of work, and a long story short, we have generated what we have called the cell age clock. So this is a clock which we believe monitors and enables the measurement of the aging of a cell. And so this is how the cell age clock performs at predicting our passage, or the passage of time essentially for our cells against the actual passage. And kind of not surprisingly, we built the clock to be able to measure this. And actually, when we validate the clock, it performs very well at actually measuring this time, this cell aging time. For those of you who like to look at the individual CPGs, this is just a heat map showing each of the CPGs that contributes to our clock along the rows. And as we move across the columns, we're moving through time. So you can see that there are changes in our clock as we move through time, and we're capturing changes that are or sites that are becoming both hypo and hypermethylated during this this transition, this passage of time. And so what we then wanted to ask was, what happens to the ticking of our cell age clock when we put on anti-aging drugs? Can we influence the cell age clock? And so we started by looking at some very well-established anti-aging compounds, rapamycin and trametinib. Really importantly for the work that we did, we wanted to be confident that we weren't just influencing the mitotic rate of the cells by applying these compounds. We wanted to be really confident that we were looking at the slowing, asking directly whether the compounds were influencing the clock, not just the rate of proliferation. So we used through a lot of kind of testing, incredibly low doses of both rapamycin and trametinib to ask this question. And these are doses which have no influence on the cumulative population doublings that we see during this period of our treatment. We did this work in all of the things that we have on this slide, but I'm just going to show you, in the interest of time, our data from rapamycin. And I'm also just going to show you this in one cellular model. So what we wanted to do was ask whether low-dose rapamycin could decelerate the cell age clock. And importantly, as I mentioned, this is uncoupled from mitosis. So what we're looking at here is the period of time that we treated the cells with rapamycin. And along this axis here, we're looking at the predicted passage. So if our values come out at 0, then what we've done is we've had no impact on the ticking of the cell age clock. Anything that is below this line has kind of slowed the cell age clock. So this is what we found with rapamycin. So actually, even with a short-term treatment, we're able to slow the ticking of the cell age clock. But actually, really excitingly and really interestingly, from the point of view of rapamycins and rapalogs, is that with a sustained treatment of rapamycin, we're actually able to, what it looks like, to reverse, not just slow, but actually take the cell age clock backwards, because these cells are much, much younger as far as the cell age clock goes than they would be if they sat up here. And so we then wanted to ask, can we use the cell age clock as a platform for discovering new anti-cell aging compounds? And so again, to cut a long story short, we did a range of different screening exercises, and we identified a number of compounds, 2 of which I'm showing here, which could slow the cell age clock, and in the case of compound 2, could also decelerate, sort of almost put the clock into reverse. We obviously wanted to ask whether these compounds were able to do anything to a range of different markers of senescence. And essentially what this series of microscopy images is showing you is that for rapamycin, we can suppress a range of different senescence markers, P21, P16, SA beta gal, the SAS factor interleukin 6, and we could also influence the structure of the nucleolin, a sort of a surrogate for the nucleolus. And then we have similar data here for our second compound. And as you can see here again, we've managed to influence a whole host of senescence markers by treatment with this compound. So then, having kind of identified some potentially interesting drugs that influence cellular aging, we then wanted to ask whether these drugs could actually influence lifespan. And to do this, we took advantage of Drosophila because of its sort of an expedited model for examining lifespan. And we used a specially formulated halidic medium, which was developed by Linda Partridge and Ivana when she was working with Linda. And what this medium enables us to do is to have an enhanced nutrients and drug bioavailability. And unsurprisingly, we find that rapamycin can extend lifespan, as you would expect. And really excitingly, both of our compounds were able to do something similar in these experiments. So the compounds which were able to decelerate the cell age clock in vitro were able to extend lifespan in vivo. So what we think we've done is we've been able to identify, define a cell age clock, which measures the ticking of cellular aging. And we've been able to use this clock as a platform for the expedited discovery of compounds which can decelerate cellular aging, both in vitro and extend lifespan in vivo. And so really what we're excited to do now is to use this platform to identify novel drivers, pharmacological targets and agents in this context. So just a few quick thank yous. I thanked most of the people as we went through, but particular thanks to Ellie, Deb, Rob, and the team at UCL, so Stefan and Ivana and Nila. And with that, I'll thank you for your attention.
Thank you so much, Cleo. Great presentation. Let me open this first Q&A session with it might be a little bit more general question. You said that you can reverse the senescent state, take cells backward in time. Mm-hmm. I think the entire field is really talking about senolytics, so removal of cells. Can you tell us a little bit what the pros and cons are of the reversal versus removal of these senescent cells?
Sure. So I completely agree. The field is very excited about senolytics. I think we're on a teetering point of whether they're going to come through for us. So I guess to answer your question about what the difference is between senolytics and the reversal. So the reversal for us really is a tool which has enabled us to show that senescence is malleable in a backwards direction. But actually what that suggested to us was that we could actually restrain senescence. And so if we can restrain senescence, what that might mean, and this is complete conjecture, is that we could slow the accumulation of senescent cells within our tissues and actually delay the point at which we might need to think about a senolytic intervention to perhaps flush out those senescent cells. And if you think about the wheel that we all know so well, if we can slow down the accumulation of senescent cells, one of the hallmarks of aging, what that might mean is that we can slow down aging itself. And so that was really our thinking. I think the kind of the slowing the cell aging clock is something that would kind of precede anything that you might think about from a senolytic point of view. Okay, that's very interesting that the compounds that we find can actually influence the epigenetic clocks in that way.
Yeah, very interesting. So you're really trying to prevent the more accumulation of damage, and there with the senescent state in the end. And I think our field is quite obviously going into direction that senescence is not dichotomous. It's not one day you are not senescent, but it's a process. And yes, I think that's a very important field. Our field, the geroscience field, is also lots talking about clocks, and especially about epigenetic clocks, which you also introduce for in vitro studies using repetitive aging cells, in the end being senescent. I think Leonard Heiflich did great endeavors and contributions to the field. Now we are applying clocks to that. I'm sometimes a little puzzled in our field that we are just looking at the sites, maybe also a little bit where they are, the methylated sites. What can we learn in your view, using your clock, about really the pathological drivers where CPG sites are methylated? So when do we really make the move, or not just counting and observing where changes are?
I think that's the elephant in the room. Because we're great at measuring these sites, aren't we? But when you try and link those sites to the biology, and you mentioned at the beginning that my lab loves delving into molecular mechanisms, so there is a big gap in our understanding as to what these sites mean for all the other omics that we do, what it means for our translator, for example. I would love to know the answer to that. We found some in the paper that we did publish a few years ago. We found some relationship between the sites that changed in senescence and the transcriptome, but what that is actually meaning for the biology of the cells, we're still trying to unravel that. And actually, I think joining those dots up is going to be, if someone can do that, that will be transformative for the field of clocks and biology. Because at the moment, clocks are brilliant at measuring stuff, but they're not maybe brilliant at telling us what's actually mechanistically happening.
Yeah, indeed, they're just biomarkers, and that can be yellow fingers. So why should we have a clock and spending lots of money in measuring the epigenetic changes? But let us dive a little bit more deeper, because if there is a CPG site being methylated and it's in a promoted region, I learned as just a clinician, you can switch on and off genes. So would that not be already a sign to look at and to deep dive a little bit more in the pathology?
Yeah, I agree completely but I think what we don't yet know is, if you took a site and you looked in the promoter, there's no way to predict whether it's going to up or down regulate that specific site. So to really answer that question, you need to go and do all the biochemistry and the biology, cell biology, to understand that. So I think our understanding of what a methylation change means, and being able to look at a site and say, okay, that's going to lead to this. I don't think we have that level of comprehension about methylation regulation yet. If someone disagrees in the audience and has a way to predict that, then please raise your hand and let us know. I think if we can solve that, and I think the more data we have, the closer we will get to being able to do that. I think that will mean a big leap forward. Okay, so I was triggered a little bit by more data. What I see in the field, if we have more data, we have even more clocks. And this is true. And they are sometimes correlated and sometimes not. So we have a BMI clock, we have a mass strength clock, we have an omics clock. I think not only one, but hundreds. Yeah. You made a clock for fibroblasts. If you would use endothelial cells or T cells, would that clock be different? If you come back to me in a bit of time, I can give you a categorical answer. I don't know yet, but I wouldn't be surprised if we need a range of different clocks for different cell types. Yeah. And so I guess it depends. So at the moment, I think within the field of senescence, we're still trying to understand in vivo, in our tissues, as we age, which cells are senescent and which cells are contributing through their senescence phenotype to aging. And one of the reasons we picked fibroblasts is because they are one of the cell types that people believe in vivo do become senescent with age, and we see an accumulation of that particular cell type in our tissues with age. I think the more data, Andrea, please, more data. The realms of multiplex are really opening up our understanding of senescence in tissues. And I think once we have insights from projects such as Senet, for example, we'll have a much clearer idea of which cells are, well, we might have a much more muddied picture, I'm not sure. But it offers the possibility, at least, of having a clearer idea of which cells are really senescent, by which criteria. And then enabling us to think about which cell types we want to really understand from the perspective of senescence in that wheel that is a very complicated collection of hallmarks of aging and factors that contribute to aging.
Yeah. From my history in living nearly in the lab and culturing fibroblasts during Christmas and New Year,
yeah.
I observed, I love fibroblasts, I observed that the inter-individual differences in a fibroblast culture is enormous. And maybe even because of the techniques we apply when reculturing them, feeding them, etc. You're making use of repetitive senescence. Have you thought about stressing these cells and applying stress-induced senescence? And while doing so, do you think, or maybe you did it, that the clock ticks differently or not?
Do you want to come back to me? Invite me in a year again. We have lots of stuff going on. We don't have the answers for that yet, but I think it's a really, really interesting question, particularly when you think about the physiological context, in that there will be lots of intrinsic triggers that could drive a cell into senescence that isn't just a replicative trigger. And so you mentioned different cell types and different clocks. We might need different triggers, different clocks for different triggers, potentially.
Yeah, and measuring the difference in senescence types, because also I think our field is now learning that a senescent cell is not a senescent cell, and hopefully these clocks will help in distinguishing what is different in these kinds of cells based on the trigger, but also on the phenotype, on deeper mechanisms. But I'm very enthusiastic. That's a little bit my closing questions to you before going to Sharar, who has lots of questions of the audience. You used rapamycin, and rapamycin, I think, is the repurposed drug being tested in a couple of groups, including ours, to see if we can reverse the aging process in humans. We are preparing the RCT at the moment. And
very exciting.
Yes, tell us. So how could your experiments in vitro in repetitive senescence, how would you advise us? Or maybe we could collaborate. What should we actually measure to see if rapamycin is able to reverse time in the cell types we might have access to? So
rapamycin, you kind of spot on. Rapamycin wasn't just picked at random; it was picked precisely because of that reason, and it was a really nice benchmark to act as a proof of principle for our screening platform, essentially. To answer your question as to what you should screen for, in an ideal world, what I would do is take your tissues, disassociate them, try and isolate the senescent cells, a bit like the smoothie analogy. Pull out the blueberries and see whether your treatments are influencing the cell age clock. And if you can, and have unlimited funds, to maybe look at some other clocks as well. But I'm quite taken at the moment by this idea of different clocks for each of the different components of the wheel. I think it's an interesting way to look at aging and to kind of tease out the smoothie, basically.
Yeah, and that I think in the end will also be the basis for personalized approaches, because it might be that one clock, proteostasis might be more announced in a certain individual or a certain organ or even cell compared to another cell suffering from telomere attrition, for example.
Yeah, precisely.
Absolutely. Yeah. So moving on to Shara Kampimpin. And I just have to announce, maybe this is the last show she is on, because she is very exciting, starting a PhD trajectory in another group. So very unfortunate that we are not losing you. You will still be at NUS. Let us give you the floor to see what the questions from the audience are.
Thank you so much. Thank you, Prof. Andrea and Prof. Bishop. That was a very insightful talk, and our audiences really enjoyed it. So we received quite a number of questions, and I think we can start off with a more technical question. So we have a question. One of our audience asked, when you're doing your experiment, have you observed if the length of the telomeres were increased when the senescence is slowed down based on the cell age clock?
We haven't looked at the length of the telomeres. It's something that we should do, and I think it would be really interesting to look at, and to look at it particularly in the context of our anti-cell aging compounds, to see whether that's influencing. Because we're not influencing the mitotic rate, that might suggest that we aren't directly influencing telomere length. But it would be very interesting if we were.
Thank you for that. So our audiences are also very interested to know whether you can reverse the cell age clock after cells have reached senescence and fully stopped proliferating.
So in the very early data that I showed in the talk, we looked at the methylome, which was a number of different sites, so that's 725 CPG sites, and for the vast majority of those we were able to reverse that process. What we haven't done yet is take deeply senescent cells and kind of reverse them and track the clock over a period of time to see whether, when we've kind of reverted the clock, whether it starts ticking again. And if it does start ticking again, at what rate it starts ticking. So have we really taken cells from a state where they've been in senescence for a period of time, we've reverted them to something that is, in many cases, a signature that is 200 days younger. Does the ticking kind of almost start again, or do we sort of revert really quite quickly back to an aged methylome? We haven't done that yet, but it would be interesting to look at.
Thank you. I think a number of our audiences are also would like to know whether is the reversal cellular or tissue specific, and does it hold true across all cell types?
It's a great question. So we have looked in fibroblasts and epithelial cells from 2 different tissues so far, and in the fibroblasts and the epithelial cells from skin and from mammary tissue, and in all of those contexts we can reverse senescence if we give the cells the right triggers. We chose skin because it undergoes a range of different physiological aging stimuli, including quite a lot of extrinsic ones. And we chose mammary because actually when you look at mammary cells in a lot of omics data, they kind of stand out, and they're quite hard to, the mammary clock, for example, is quite tricky to align with other tissues. But we haven't looked in other tissues, and we haven't looked in other cell types. Such Andrea mentioned T cells. I think we haven't done anything with T cells.
I think some of our audiences are also very curious because right now there's many different areas of many different pillars of aging. So they were just wondering whether if we are just looking at DNA methylation as a basis for discovery of novel compounds, is it possible that we might potentially miss a group of compounds that can positively affect other hallmarks of aging?
Yeah, of course. Certainly. I guess we took this approach because by defining our cell age clock, which has got 42 different sites in it, what we're able to do is to take a reasonably high throughput approach to compound screening. But yeah, the audience is absolutely right. This approach won't capture everything, and we could well be missing things that are influencing histone epigenetic marks, that are influencing microRNA signatures, no RNAs. Yeah, the cell is a wonderful, complex universe, and we're just looking at one aspect.
We'd also like to know how do the compounds identified using the novel drug discovery platform affects other senescence markers aside from the DNA methylation?
So we've looked at a range of sort of a panel, we call them, of senescence markers. So we've looked at the effect of the compounds on cyclin-dependent kinase inhibitors P16 and P21, and we find that the compounds can suppress the levels of these proteins. Which is when Andrew and I were talking, is sort of senescence isn't a kind of a switch that the culture goes into, sort of a gradual process. We're able to suppress features of the senescence-associated secretory phenotype. So I think I showed some data for interleukin-6. We're also able to suppress SA-beta-gal, another classic marker of senescence, in vitro and in vivo. And we're also really interestingly able to influence nucleolin, which your audience is probably aware of the data looking at the nucleus in aging in tissues in vivo, and actually how that's a really intriguing and exciting biomarker of aging that does seem to kind of change. So we looked at a panel, and our compounds were able to influence each of those. And that was one of the criteria that we used to select our compounds to take them in vivo into the Drosophila model.
All right, thank you for that. I think we have one last question. I think one of our attendees was wondering, do we know the optimal number of senescent cells that won't negatively affect overall health during aging? Or is it the general consensus that all senescent cells during aging is bad? I think a lot of people are very concerned about senescent cells right now. Yeah.
The short answer to the question is no. we don't know. Well, I don't know. Again, if somebody in the audience knows, please pipe up. I think we're still trying to understand what senescent cell burden actually means in vivo. And we're still developing the tools and the techniques that we need to accurately detect these cells in vivo. There's a lot of really exciting stuff going on around being able to detect senescent cells in vivo. You're probably aware of a lot of the fluorometric and vesicle encapsulated dyes that people are starting to use to try and detect senescent cells through MRI, for example, which I think is really kind of a really fascinating area of our ability to detect these cells. But I think at the moment, if you asked me how many senescent cells we have in our bodies at different times, I think that's, well, individual specific, but also I don't think we can accurately quantitate that yet. But I think if we get to that point, being able to accurately measure senescent cell burden, I think it's going to be very interesting to see how that can be used as a biomarker to predict our trajectories for different aging diseases. Andrew and I spoke earlier about different senescent cell burdens in different tissues. If you can imagine a scenario where we can take a little probe, have an MRI scan, can see, oh, Cleo, you've got a lot of senescent cells in your kidney, I don't know. That might mean you're on this particular health trajectory. But actually, if you've got a lot of senescent cells in another tissue, it might mean that you might consider a different health management strategy for that person. So that was I gave you a short answer, which is no, and I gave you a lengthy answer, which is just some of my musings about where we might be going in the future.
Perhaps we can take one more question. I think somebody asked, whether are the cells raised under a light dark cycle? And if so, how many hours of light and how many hours of dark per day? I think there's a more technical question.
You know, I think the interplay between the different components of the aging hallmark wheel, I think, are very interesting. And a decay in our ability to maintain our circadian rhythm is something that happens as we get old. I'm always very keen that my lab do their tissue culture at pretty much the same time of day, whatever time that is for their cells, so that they get, it might sound a bit new age, but so that they are in a rhythm. I actually think that's really important. And if you look at cells that have been in culture for a long time, you can still see the cycling of circadian proteins in the cells. So that is, I think, is really, really fascinating. Did we use a light and dark cell cycle? Did we use a light and dark cycle for our cells? No, we didn't. They come out for their tissue culture normally in the morning. So they get a short burst of light, and then they're back in a dark incubator for the remainder of the time. But I think that connection is fascinating, and there are a few papers starting to look at the link between senescence and circadian rhythm, which I think are really interesting.
Yeah. Okay. Thank you.
Okay. That was great. So you are not only taking well care of your cells, but it seems also taking care of your arrays, etc., and PhD students, that they have enough sleep, which is great, because there's one study looking at the number of senescent cells in blood and the sleep quality. So well done. Thank you so much, Cleo. So join me in thanking Professor Bishop for this very nice presentation. And once again, she is from the Queen Marie University. And also, I think we should give an applause again for Reef from the Temasek Polytechnic, who had the short presentation. You can leave any suggestions in the panelists and all attendees options. Please do so, because it's highly valued what you think and what we should do, because we would like to serve you. Please follow us on LinkedIn, on Twitter and Instagram, etc., and enjoy our recorded webinars, because they are available. If you missed one, of course you shouldn't, but if you missed one, they can be found on YouTube. The next episode is on November 3rd this year, in 2022, and Professor Brian Kennedy will welcome Ivan Vetchkov, and he is the chairman of AdFixes. And I had to look it up. It is a private investor company focusing on fintech, health tech, and edtech. So you will learn what edtech is. I am closing this webinar showcasing a colleague of mine, also an internal medicine specialist. Take care.
My name is Dr. Melissa M. Freeman. I have been in the practice of internal medicine for more than 60 years. Could I have her chart, please? And I am 92 years of age.
Dr. Freeman owns and operates this private practice in Harlem.
How are you? Hello.
A business she's run for more than 37 years.
What's a typical day for you, Doctor?
Well, I start in the morning working in my drug clinic, and I complete that, and then I take transportation and come here to my office.
Now, she doesn't shy away from public transportation. She embraces it, traveling on the New York City subway at least twice a week.
I'm a New Yorker, traveled by subway, and if I have to get somewhere and the subway is the best way to get there, I will board the subway and take it.
But I hear, like, you keep up with everybody. You're tackling the steps.
Well, that's the only way you can get to the subways.
No plans to retire?
Not that I can see in the near future, no.
What is your secret to not just your longevity, but your vitality?
You do what you have to do. You have to move on. As long as God gives you life and gives me strength, I have to keep doing it. And he has been very, very merciful for me.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.